Okay, so here we are. So in the last video, I kind of introduced you to the idea of the action potential, and that this action potential, this spike, is an electrochemical impulse, which is the basic unit of information used by the brain. And it's the unit of information that is actually generated by neurons. It's the fundamental function of a neuron is to generate these action potentials. So in this video, I want to spend quite a bit of time talking about how the action potential is constructed. Why is there an action potential? Because it's of absolutely fundamental importance. Okay, so let's go back to this diagram. So here we see the action potentials you see before. Again, just to reiterate, why not? Can't do any harm. The membrane potential, when the neuron is kind of doing nothing, sits here. The rest potential. Then when an action potential is initiated, and in this video and probably the next, we'll talk about what initiates it. It shoots up very, very rapidly, past 0 up to about plus 40, and then comes all the way back down, overshoots a bit, and then goes back to the resting potential until it fires again. And it might do that maybe, in some of the fastest firing neurons, that can happen up to maybe 250 times per second. Okay, so let's talk about how the action potential is constructed. So in order to do that, so this is where, okay, back to the camera. So this is where these kind of prerequisites come in particularly the idea of ions and the idea of concentration gradients. So we're going to look at, in particular, the membrane potential first of all, and what that is. I said that the action potential occurs when the membrane potential goes from kind of minus 70 millivolts, shoots up to plus 40, and then comes back down again. What does that mean? So in this video, we'll cover exactly what I mean by that. Okay, so let's go to the board and look at how all this works. Okay, clean board. So first of all, we will let's draw a membrane. So a couple of lines should do the trick. Splendid. So this is your membrane. This is, in fact, you'll know, the plasma membrane of the neuron, and we will say this is inside of the neuron, and this is the outside of the neuron. Now, in neurons particularly, in all cells there are ions, but what's special about neurons is that there is this imbalance between ions inside and outside of the cell. So we'll talk about 2 ions in this video, but we'll introduce another ion as well. So why not do it in yellow? So we'll talk about sodium ions. So sodium, of course, is Na, which carry a positive charge, and potassium ions, which I will do in, I don't know, orange will do it. Potassium ion, potassium, of course, which is K, and you're going to find potassium ions and sodium ions both inside and outside of the cell. However, and this is the key point, in a neuron, what you always find, is that there is a very high concentration of sodium ions outside the neuron and a low concentration of sodium ions carrying positive charge on the inside. For potassium ions, however, this situation is reversed, and we have a high concentration of potassium ions on the inside and a low concentration or lower concentration of potassium ions on the outside. So already your mind should be thinking, oh, this is a concentration gradient. It is. It's not just a concentration gradient, it's also an electrical gradient, right? Because you've got more positive charge, more positively charged sodium ions on the outside. Now, in fact, if you want to kind of get the total charge carried by sodium and potassium, you simply have to add up the charges. And what you find is that there is a higher number of positive charge in total on the outside of the membrane. So we would say, just write this as high positive charge, and a lower number of sodium and potassium overall. So even though there's a higher concentration of potassium on the inside, overall the total positive charge on the inside is lower. So if we take the number of positive charges on the inside, so on the inside, and we subtract, okay, not really much space, the total positive charge on the outside, that is going to equal a negative number, simply because there's more total positive charge on the outside. And this is where that resting membrane potential came from. You remember the action potential started, look back at the camera here, so the action potential, remember, started from a negative level. So the way that this imbalance of charges is measured is using voltage. That basically simply measures the uneven distribution of charge, what's called the charge separation from between the inside and the outside of the neuron. So what we say is that overall, we say that a neuron is negatively charged on the inside. And that's simply because of the uneven distribution of positive charges, with more positive charge on the outside overall and lower positive charge on the inside. Okay, good. So let's go back to the board and let's kind of formalize this a little bit better. So we'll clear this. So what do I want? What do I want? What do I want? What do I want? I want a line. Let's use a line here. So now we can draw a nice graph. And remember, so this should ring a bell. Ring a bell. People still say that? I think so. It's reasonable. So we had 0 here. And if you recall, the neuron, when it wasn't doing anything, had a membrane potential kind of down here, which was minus 70 millivolts. Again, millivolts is just the unit of measurement used to measure that separation, charge separation. So now you know why that sits there. This is called the resting potential. And all neurons, and this is again a slight simplification, but we'll talk more about this in a later unit. When a neuron is doing nothing, the neuron is resting, its membrane potential will tend to sit around minus 70 because of this charge separation. That's not always the case, and again, that will become important later. So what's happening then with the action potential? So let's draw an action potential, so you're now familiar with what an action potential looks like. I think an action potential should be red, shouldn't it? Of course it should. So the neuron membrane potential here, we've got time here. the units of time aren't important. So the neuron sits, so over time, neuron sits, resting potential, doing nothing, and then for some reason, suddenly the membrane potential jumps up to around 40 millivolts, and then just as quickly comes back down, overshoots a bit, and comes back down. So what have we learned here? So the action potential is a reversal of the membrane potential. The membrane potential goes from being negative because of this uneven distribution of positive charges to being positive very, very briefly and then comes back down again. So this is the way the neuron generates information. It's by rapidly flipping its membrane potential from negative to positive and then back to negative again. So the question then is, how is that action potential generated? Or in other words, what makes the membrane potential suddenly flip to positive and then back to negative again? So to explain that, we need to think about channels. And channels are what ions use to get through the membrane. Now, if you remember from your biology class about the structure of the plasma membrane, it's fatty, which means or hydrophobic. It doesn't like water, like a rabid dog. Remember Cujo? Oh, gave me nightmares, that did. Ugh. Anyway, Stephen King. Anyway, Cujo's put me off. Anyway, yes, so the member I was talking about. Oh my God, I just think that dog, the saliva. St. Bernard, wasn't it? St. Bernard. Such a sweet dog that's like bit by a bat. Bats, stay away from bats, clearly. We've learned that, haven't we? Yeah, so what causes, yes, ion channels, that was it. So normally, because the membrane is fatty, it doesn't like water. That was where I got to Cujo. And it doesn't like ions. So ions can't get through the membrane. This is actually how, because of course, if you think about it, the sodium ions, because they're imbalanced, high on the outside, low concentration on the inside, they want to move down that concentration gradient, don't they? But they can't because the membrane is in the way. That's what keeping the charge separation there. So in order to reverse the membrane potential, in order to initiate one of these spikes, we need to introduce the idea of channels. And in particular, a special type of channel, and a channel is a protein, a special type of channel called a voltage-gated potassium channel and voltage-gated sodium channels. So let's look at these. Let's draw these, see if this makes sense. Okay, so again, we will have our membrane. So here is our membrane, thus. And of course, it is a fatty membrane, so we might even draw, there we go, illustrate there. So normally, sodium ions, they're unable to get through the membrane, and the same applies to potassium ions as well. I have some on the inside as well, like this. But a channel is a type of protein that, as you might expect, I think, let's draw a voltage-gated sodium channel first. So a sodium channel is simply, if you kind of slice through it, it would kind of look like this-ish. Now this is a channel that is in a closed conformation. So, so channels are actually quite complicated, but we will keep it simple and say, let's get rid of potassium for the time being. We can. It's just confusing us. There we go. So this channel is a gated channel. So gated means it has a gate. In other words, the gate can be open, the gate can be closed. If the gate is closed, sodium cannot get through. No. However, the channel can also be in an open form. So when the gate is open, like this, and the sodium ions can then pass through. Now, this is the key point. What determines whether a voltage-gated sodium channel is open or closed? Well, the clue is in the name. It's gated by voltage. So, let's go back to our drawing of the membrane potential. So you'll get used to this graph after a while. We'll be using it for a while. So we have our usual points. Here we go. Here's our 0. And of course, you're all now familiar. This is our resting potential. We'll just abbreviate that to Rp. Now, sodium channels, they open at a specific membrane potential. So when the neuron is at its resting potential, so it's sat at kind of minus 70, these voltage-gated, draw this. So these voltage-gated sodium channel, voltage-gated sodium channel is closed, which means sodium ions cannot travel through the channel and cannot travel down that concentration gradient. However, and this is where it gets interesting, and I'll do this in yellow, it's so important, there is a particular membrane potential, a particular voltage at which these voltage-gated sodium channels open. And that is around minus 55 millivolts. So if the membrane potential, for some reason, which we will get to, if the membrane potential for some reason, say it's moving up and down, reaches this minus 55, then the voltage-gated sodium channels will open. Now, let's think about that. So, what would happen if suddenly all of these voltage-gated sodium channels opened? Well, you need to think about the concentration gradient of these of the sodium ions. So we know that this high concentration of sodium ions on the outside of the cell and a low concentration on the inside. So if suddenly all of these voltage-gated sodium channels opened, what would happen to the sodium ions? Of course, they would suddenly start flooding in down their concentration gradient through the channels and into the cell. And that is indeed exactly what they do. However, can we then explain or then think about what's going to happen to the membrane potential? Now, remember, when we first introduced the idea of the membrane potential, the idea was that it's the imbalance of positive and negative charge which generates the membrane potential, this charge separation. And there is more positive charge on the outside compared to the inside, which gives the neuron, the inside of the neuron has an overall negative charge compared to the outside. So what happens when positive charge starts, i.e. sodium ions, starts flooding into the neuron through these freshly opened voltage-gated sodium channels? Well, the membrane potential is going to go up. It's going to go more positive, right? So we can see this, draw this on the diagram. Voltage-gated sodium channels, they open here. The membrane potential suddenly shoots upwards. What is that? That, of course, is the first leg or the first arm or a leg of the action potential. Now, of course, this will keep going up and up and up, in theory, until there is the charges, the concentration of sodium. It's actually a little bit more complicated than that, but you can imagine it would keep going up until the concentration of sodium is balanced, right? Until it reaches an equilibrium. But actually, something interesting happens around about this point here, when the membrane potential reaches about 40 millivolts. What do you think happens there? Well, the sodium channels actually start to close. So they're not open permanently. They actually start to close again. So they open briefly to allow sodium in, and then they close. But then something else happens. Another type of voltage-gated ion channel opens. This is the voltage-gated potassium channel. So here, at this point, voltage-gated potassium channel open. Now, what's going to happen there? Well, let's think about it. We know that the sodium channels, the voltage-gated sodium channels have closed, so sodium has stopped flowing into the cell. And now these voltage-gated potassium channels have opened. Now let's think about the concentration gradients involved here. So again, there's a concentration gradient between the inside and outside with potassium ions, just like there is with sodium ions. However, it is in the opposite direction. So there's a higher concentration of potassium on the inside, so potassium ions flow out. So this time, in the opposite direction to sodium. So potassium ions flow out, takes positive charge out of the cell. What happens to the membrane potential? It comes back down again. Goes all the way back down. It will actually overshoot a bit. And then there are systems, again, the potassium channels will then close again. And then there are these special pumps that will kind of restore everything back to the resting potential. So that is the action potential. That is the mechanics, the basic mechanics of the action potential. When the membrane potential reaches around minus 55 millivolts, voltage-gated sodium channels suddenly open, sodium rushes in, membrane potential shoots up, goes up to about 40 millivolts, then the sodium channels shut, potassium channels then open, potassium rushes out of the cell, taking positive charge with it, and the membrane potential shoots back down, overshoots a bit, and then it settles back to its resting potential. That is an action potential.